my name's John. I'm from George Mason University. I'm in the Lim Lab of Evolutionary Genomics. I'm a master's student just finishing up there. And I'm going to talk to you guys a little bit about the population genetic structure of what we think is early stage parapatric ecological speciation in a subspecies of song sparrow here in eastern North America. So just to start out, in case you don't know anything about the song sparrow, it's this amazing North American songbird that has this incredible range of phenotypes. It's found basically in every terrestrial habitat you can think of across North America. So here in the east, we sort of had this widespread generalist that you can find outside this building. But you have extreme examples of subspecies in the Aleutian Islands that are very large, subspecies that live in these really hot deserts in the southwest. And basically anywhere you go, you can find a really cool subspecies of song sparrow. So there's 25 subspecies currently described. And obviously, they've been interesting people for a while because of their extreme diversity. So there's been lots of work before looking at how this morphology within the species varies according to the habitats which they occur in. So it's led people to think about them in this kind of ecological divergence process for quite a while. So just a little bit of background about what we know about the genetics of the song sparrow. Some folks set out to sequence the mitochondrial control region a while ago, and they surprisingly found that despite this incredible phenotypic diversity and this giant range, there is no structural variation in mitochondrial genes across the entire continent. So an individual in Alaska might have the same phenotype or same genotype as an individual in Mexico. And so that kind of begs the question then, well, where did all this diversity come from? I mean, obviously there's divergence between populations because of the extreme phenotypic diversity. And so normally we think about speciation or divergence in this kind of allopatric context where you have a clean break and that gives time for neutral loci to sort of gradually diverge. But that's obviously not the case most of the time, and it can be this more messy process where you have primary divergence without a clean break. And so it can be a little bit harder to study, especially before genomic technologies that we have today. So we're thinking again about this species in the sense of parapatric ecological speciation. A lot of these subspecies abut each other, and they intergrade where they co-occur. And so we imagine that there's probably some ancestral population of the song sparrow that's expanded post-Pleistocene. And when it's encountered these new different habitats, it's been able to diverge in this kind of clinal pattern, which is sort of what we see today, to the extent where they're starting to separate into two lineages. And you can imagine that given enough time or extreme enough natural selection, you might end up with a whole bunch of different subspecies of song sparrow, or species of song sparrow. So that brings us to our system. Here in eastern North America, we have two subspecies of song sparrow, which is less diverse than in the west, where they have dozens. But along the eastern North America, we have this salt marsh and sand dune habitat along the coast, which is shown here in pink, and then it looks like that. Not a great place to live if you're a songbird. Not a lot of water, very hot. But we also have all this nice suburban habitat, right? This kind of secondary successional stuff just everywhere. And so in this secondary successional stuff in suburbs and things like that, we get this subspecies eastern song sparrow, Melospisa melodia. And again, you can find it pretty much anywhere in eastern North America that you have this kind of anthropogenic habitat. But in along these coastal sand dunes and in these salt marshes where it's not so great to live, you get kind of a cryptic subspecies. It doesn't look super different from the eastern song sparrow, but it is this ecological specialist that you only find in these sand dunes and is morphologically diagnostic. And they kind of illustrate Allen's rule, so some prior research efforts really looked into this a lot in this system. And basically, Allen's rule being that if you live in somewhere that's hot and water limited, you tend to have proportionally larger appendages. And so the Atlantic Song Sparrow along the coast has a much larger bill, is the main morphological distinguisher, and the bill can be used to radiate heat. So just talking about that a little bit more, some people were really interested in what the selection differences are in this system. So if you take a bird and you put it, these are actually song sparrows, if you put a song sparrow in kind of a cold environment, you'll notice that they lose heat through the bill, so that's a disadvantage if you have a large beak in a cold environment. But at room temperature, pretty comfortable, and then as you heat them up, they're able to use the beak as this dry radiator of heat, so they're not losing water when they're trying to transpire heat. And so there's an advantage to having a big bill in a hot water limited environment, while it might be a disadvantage to have that somewhere where you can get out of the sun into the shade and you have plenty of fresh water. Just again, they kept going down this rabbit hole, so if you look at the inside of the bill, there's actually more surface area if you have a bigger bill, which allows you to prevent losing water through breathing. So again, it's adaptively advantaged in a song sparrow to have a bigger bill if you live somewhere water limited. And also in the eastern song sparrow, even if you control for the size of the bill, they have more surface area on these interior respiratory nasal turbinates. So there's really this pressure on them to have these morphological traits that make them specialized to their environment. So just to kind of summarize all the prior work that's been done proving the selection between these two systems, you could imagine you have bill size variation in the song sparrow in eastern North America, and then you could plot your ability to conserve heat as diametrically opposed to your ability to radiate heat, and you would get a function kind of like this, right, where if you have a really big beak, you're really good at radiating heat, and if you have a really small beak, you're really good at conserving heat. And so it just creates this sort of ecological barrier to gene flow between these two subspecies, despite the fact that they abut each other. Where you want to be on this side of the line if you're an inland subspecies, but if you're an inland habitat, but in coastal habitat, you want to be on the other side of the line. And certainly the bill isn't the only trait that might follow this pattern, it's just external, so we're able to really see it. But you can imagine osmoregulatory functions or other things might follow a similar kind of divergent selection between these populations. So that's what we know. But we don't really know anything about the genomic basis in this system of this divergence between the two populations. So we decided to set out and try to look into it a little bit. So we said if the eastern and Atlantic song sparrow are a recent parapatric divergence due to this ecological pressure, we would expect neutral genetic markers to not be useful differentiating them, which would either be due to interbreeding and or because of rapid differentiation. They didn't have complete lineage sorting. And then conversely, we expect that loci under ecological selection in this case would be useful for differentiating them because that's what's underlying these divergent phenotypes. And it might be, for example, limited to genomic islands of differentiation, which I'm sure everybody's been to a talk that's talked about those today. So the samples that we have were collected by some former research efforts by folks at the Smithsonian Migratory Bird Center, and some came from the Smithsonian Natural History Museum. And they represent this kind of ecological gradient from this coastal environment across the coastal plain and then into these inland environments, and even some extreme inland ones here in western Maryland, and off screen over here somewhere we've got some samples from southwestern Virginia. So pretty broad transect that goes across this ecological gradient. And we've got a pretty robust sample size thanks to these former research efforts. We've got 200 and some odd DNA samples that were just sitting in a freezer. So, powerhouse of the cell, we started out with mitochondrial DNA. Just to see what would happen, because why not? So we took a subset of 48 individuals, and we amplified and sequenced the ND2 gene instead of the control region, just because that's already been done on the continental scale, just to see and prove that maybe it's not useful for telling these subspecies apart. So just to give it some statistical power, we performed an AMOVA, we also made a minimum spanning network, and then we also performed a Mantiel test. And so the results of that are, as we expected, there's really no clear structure. So the blue are subspecies from inland on the mainland of Virginia. The gray are these peninsular individuals that we consider the eastern song sparrow, but they're close to, so they're proximal to the habitat of the Atlantic song sparrow. And then the yellow is the Atlantic song sparrow on the coast. And so you can see here the AMOVA, absolutely insignificant results there, and the Mantiel test shows that there's absolutely no geographic pattern here, just like we expected from what's already been done in song sparrow mitochondrial DNA. So the conclusions from this are consistent with what's been found in the control region, like you'd expect, and so this tells us that the populations diverge recently and or they regularly interbreed, which would be consistent with this parapatric ecological divergence. So then we went to RADseq, everybody's best friend right now. We did a variant called 3RAD by Travis Glenn that's fairly new, so we kind of tested that out, and it worked pretty well. So after filtering in stacks, we still had our 200 some samples. We had to exclude some because of poor sequence quality, but we still had 163 individuals, so a nice robust sample size. You can see they kind of represent our different habitat zones pretty well still. And then I just have this picture of me smiling with my advisor over here because we got lots of loci and lots of SNPs, yay! So we have 11,700 rad loci and 70,000 SNPs after we did this 3RAD process. So recommended, it worked great. And as you do, we started out with the PCA just to see what was going on here. So again, just to kind of, it's mirrored geographically, but you have these purple sites, or these distal inland sites from western Maryland and then somewhere off the screen in Virginia. You have these blue sites, which are inland, but they're kind of proximal to the coast. You have these green sites, which are peninsular, so they're eastern song sparrow putatively, but they occur near the coast. And then you have these actual coastal individuals, which should be Atlantica. And you can see you get this kind of clinal pattern here along PC1, so the variance isn't as strong as we'd like it to be, but there is some kind of differentiation here. And just because if you can see it, I'll talk about it a little more later, but you can also notice there's this huge variation within the coastal individuals, which is kind of strange. But so PC1 kind of gives us some information that we can use. So let's zoom in here. What we're really interested in, again, is this kind of contact zone between these eastern song sparrows in this region and the coastal individuals here in these transitional habitats, right? So if we just zoom in on the PCA there, you can kind of see this pattern, right, where you have this clinal shift going from the coastal individuals in pink are split from the mainland individuals in blue with this sort of transitional phase along the peninsula. So again, that could be this primary divergence, sort of pulling these two populations apart. But we can't really be sure because you could also see that pattern from secondary contact, so I don't want to over-conclude from it. So then we did a structure plot, standard stuff again. And you can see here, essentially there isn't clear splitting, right? So if we do k equals 2, we don't get like these nice separated blocks between the coastal and the distal inland. The distal inland is kind of its own, being the really far away from everybody else. They are their own block essentially, but then all the individuals kind of proximal to the coast shown here, these blue, green, and yellow, they kind of are this slow transition into a coastal ancestry is what we're calling it. And k equals 2 seems to be the best supported, but also if you just set k equals to 3, there's an interesting thing here. You would expect that the individuals that would separate at k equals 3 would be the ones from Maryland and the ones from Virginia, despite being from the same habitat. They're really far apart from each other, and they actually still cluster really well together. But in the coast, we see this kind of weird break start to happen. So yeah, interesting stuff. So just another way to visualize it, basically, if you split them into these different categories, you can see here this transition. And if you plot it geographically, so again, this is just the assignment to the coastal cluster. If you plot that against the distance from this coastal habitat, you can kind of see this clinal relationship where these guys are really definitely their own sort of group. There's some transition going on here, and then there's a lot of genomic spread going on over here. So just to come back to what I said about the PCA, there's this cluster here. These are also, when k equals 2, they're the most pure coastal type. When k equals 3, they start to break off into their own group. Just so much that these 3 orange guys here are actually the 3 outliers in PC1, 2, so there's a nice correlation there. And what that might be a result of is it's actually the only ones that were really purely coastal genotypes were from Assateague Island here, which is, unlike most of our areas where you kind of have this coastline, like you would find the coastal subspecies, and you have this intermediate habitat over here. It's just a sandbar with ocean and salt marshes. So we think that maybe there's just purifying selection going on there, and you can't be an intermediate, so you're weeded out. That could potentially be causing that pattern. So just to really quickly, our conclusions are that we kind of got some weak differentiation like you would expect. There's a clinal relationship in genetics using, we can see in the PCA or in the structure plots. And then there's the potential that anthropogenic change might be facilitating gene flow if they are in secondary contact, just because as humans have changed the landscape next to the coast, they might be facilitating that eastern subspecies moving in and getting closer, but we're not really sure. So future steps, we really need a reference genome for the song sparrow, surprise. Then we could do a genome-wide scan for genomic islands, like we suspect is what differentiates these populations. We could potentially even compare modern and historical intergradation if we went into museum samples and we sequenced UCEs or something. So that would be an interesting thing to see if there was more of a split before than there is today, or if this pattern just has been persistent through time. And yeah, we could look for genes for calcium channel. Anyway, thanks. I'd like to thank my funding sources, my advisors, the Smithsonian for all these samples, and thanks for hanging around. So.